Yes, thank you, John, for the invite and for the nice introduction. So I can skip my first title slide and go straight into the telling you what this MAC1 is. This is part of this big NSP3 multi-domain coronaviral protein that has multiple domain structure, and MAC1 is here at the terminal side of the protein. It falls into something that we call macrodomain, and macrodomains are widespread in organisms of all kingdoms of lives in different species. And in proteins, in human systems, they usually act as dearylating enzymes, so enzymes that reverse this post-translation protein modification that we call ADP ribosylation. So now we discovered this basic activity 2016, but still I'll say immediately that we don't know physiological targets of this protein and forward reactions that make it, but it will be clear as I go through my talk. Now for you in the audience that don't know what ADP ribosylation is, I prepared a couple of slides to help you out. So this is, as I said, post-translation protein modification, but also in recent years we discovered it can also target nucleic acids. It was actually discovered a long time ago, 60 years ago, but for multiple reasons, we still know very little about it. Part of the reasons that is very technically challenging modification to study, but secondly, for most of the decades since it was discovered, it was only looked at or mainly looked at in the light of its role in DNA repair and chromosomal structural regulation through studies of PARP1 enzyme that makes it. But now we know that this ribosyltransferase transferase enzymes that can make this modification from NAD like PARP1, they're much more widespread. And they're found in organisms from all kingdoms of lives. In mammalians, they control most any process. So it's basically general modification, as you would see, phosphorylation or ubictylation. So with this growth of complexity of pathways that are regulated by this modification, the number of enzymes was growing. And so in the last 10 years, we got aware that the largest family in humans of heritable transferases are these PARPs. There are like 17 members, but there are other families like RTCs. Well, most of these enzymes actually know very little about, but as you'll see through my talk, and I'll be telling you mostly about freshly published or unpublished data, that rate of discovery is becoming really fast and exciting. So now with the complexity of PARPs and other ribosyltransferases, transferases, we were realizing that there are different kind of type of linkages that might be linked to the protein by this modification. For example, in DNA damage response, serine is the primary target, but glutamate is probably the main thing in the relevant pathway for infection immunity that I'll talk today. Also, in some pathways like in DNA repair, this modification consists of repeating units, which we refer to as polydepurylation. And again, as you'll see in antiviral PARPs, the monarbusylation is the form that is being synthesized. So again, so here I'm going to start focusing on what is more the interest of this audience. So I'll tell you within human PARPs, there are like several PARPs that has been obvious for many years that they are highly interferon inducible, like PARP7 or PARP9 to PARP14. They also exhibit very fast evolution, so they change a lot between already different people, but between the people and primates. So it was already clear early on that there must be some sort of antiviral enzymes. And now more recently, it became apparent for most of these PARPs, there are already data that are important to suppress in some way, although it's not known how, viral infection, and it was shown, for example, for HIV, Ebola, influenza, and finally coronavirus. One special case among these PARPs is PARP7, which is interferon inducible, but actually acts as proviral activity, as shown in a mouse coronavirus model. And it is known that somehow dampens interferon response. So in its own right, this could be a drug target for an antiviral treatment development, in theory at least. But what I'm going to focus further today is this cycle of hybridization where MAC1 becomes relevant, at least to what we know, and this is its relationship with PARP called PARP14 that's conserved in most of the metazoan species actually. And what we know from genetics, almost exclusively data, that PARP14 is the main anticoronaviral PARP. So basically, if you don't have it, you have most obvious effect compared to any PARPs in terms of preventing somehow SARS-CoV-2 replication or progression. Although again, mechanistically, we know almost nothing. And so basically, our main focus of my lab is really to understand exactly mechanisms of that. So what PARP14 is targeting, is it endogenous protein, viral protein, or even nucleic acid, and how it then translates into suppressing the virus. But then again, all coronaviruses, many other viruses actually utilize macrodomain type of protein, and in coronavirus we call it MAC1, that somehow, again, that we know from genetic approaches, that it somehow counteracts PAR14 activity. So basically, if you have viruses deficient in macrodomain, just putting a point mutation in catalytic residues, these viruses won't be able to progress, in fact, in vivo models. So how does it go? Again, I can tell you immediately we don't know exactly, but I'll tell you what we know and give you this, biology context for these reactions, and then I come at the end into our efforts to develop inhibitors again, MAC1. So again, what do we know about PAR14 at the moment is we've known for a while it is the largest PARP. It's 200 kilodaltons, and until recently we didn't know almost anything about any of the domains that build these protein domains. And from, as I say, its functional relevance, in addition to antiviral activity, it's known that it somehow modulates inflammatory responses. PAR14 inhibitors are in fact even in clinical trials against different allergic inflammatory diseases, but it's again, molecular basis, it's unknown. So now, as most of the PARPs, catalytic domain is at the C-terminus of these proteins, and through our studies, biochemical studies with recombinant protein, we can at least say at this stage, this protein would be specific for modifying other protein targets at glutamate residues by monohybridization, or ends of DNA or RNA at the phosphate groups at the ends or breaks. So basically, if you offer this type of substrate, PAR14 will try in vitro to transfer ADP units on them. Apart from that, there are some other domains that might teach us what this protein might be doing, and recently identified a set of 8 cage domains in these proteins that are known from other proteins, should be some sort of DNA or RNA single-strand RNA binding domains, probably in sequence-specific manner, and modeling actually of piece of RNA or DNA nicely models along these repeats on AlphaFold model of human 14. So we don't know whether nucleic acid in this case might be relevant for activation or modification of the nucleic acid, but again, it does help us to focus our thinking what this protein might be doing in vivo. Apart from these domains, I would also just note this triple, again, macrodomains. Again, I already introduced you macrodomain as something that is held, that builds the MAC1 protein. And in fact, the first macrodomain of these 3, these 2 are just binding domains, it seems, from studies so far. But first macrodomain of PAR14 is actually the most closest macrodomain of all to MAC1, suggesting that first, probably virus at some point hijacked this gene, part of the gene, and now using this function probably against the human host. And secondly, what we know so far, again, from biochemical studies, that biochemical activities of PAR14 macrodomain 1, MD1, and MAC1 are identical. So basically, they both would reverse hybridization from glutamate or phosphate-linked substrates on nucleic acids. So just to support this, I won't go too much details into techniques for the sake of time. This is very recently published. We can make radioactively labeled substrates on proteins using modification reaction by power 14, the simplest reaction by auto modification of this of the transferase itself. And you can see that recombinant MAC1, similarly to power 14 macrodomain, remove the substrate decarboxylation of the auto-modified protein, but not the other two macrodomains. And similar situation is when you look at adenylated amino acid, nucleic acids. You can see activity for power 14, MD1, and SARS-2 MAC1. So altogether, you know, power 14 seems to be so far the only known PARP with both transferase and reversing activity in one polypeptide, suggesting that really this protein wants to tightly regulate its own activity in the cells. But then again, what I already suggested, that MAC1 seems to have hijacked this domain at some point of mammalian evolution and revert it to counteract power 14 signaling as it suits the virus. Yeah. Just a little bit going into more cellular model, just to convince you that this is a relevant pair, not just from genetic studies, but also from functional studies in vitro and in vivo. So in this model, we are using 290T human embryonic cells. And these cells are naturally one of the rare cell lines devoid of PARP14 expression. But when we introduce a PARP14 wild type gene and ectopically express it in the cell line, you can see some increase of hydroxylation in these conditions. We can detect hydroxylation here with specialized antibodies that detects any form of hydroxylation linked to a protein. But you can see when we use PARP14 macrodomain MAN mutant that can't demodify its own modification, have dramatic increase in the signal in cells. And importantly, then we can show that SARS-2 macrodomain co-expression of it can counteract this signal, and we can't see it with most of the human macrodomain proteins, such as macroD2, TARG1, and PARG, but we can see it with another one, which is macroD1. I won't go into that today. So altogether, yeah, we can show that biochemically PARP14 and MAC1 are the pairs for on and off activities, and actually that PARP14 itself has its own negative regulation by reversing its own signal. Now, moving one step up to show you a situation in endogenous situation, more close to the reality, and this time I'll show you immunofluorescence data in living cells. We are using here A549 cell, and that's quite common to study this type of pathways. So you'll see again we are using specific antibodies that would pick up any form of hydroxylation, and you can see that after interferon induction, that PAR14 is by far the major inducer of some sort of signal of hydroxylation that specifically happens, I mean, mostly around the nucleus, probably in some sort of membrane compartment. We don't know yet. We are looking at this in more details, and it nicely co-localizes with the localization of PAR14. And you can see, for example, that in PAR14 knockout cells, most of this signal of hydroxylation disappears. Now, again, to make a further point, how this works as a pair in this system, that if we co-express MAC1 again, in this kind of system, that we lose these hydroxylation signals around the nucleus. So again, we now understand this biochemistry, but I have to say we still don't know physiological consequences, exact protein or nucleic acid targets, and how does it translate in different physiological effects of, I mean, PARP14 hydroxylation activity and its reversal by MAC1. So these consequences in this warfare are surely not clear, and I hope next time if we talk in a couple of years I'll have something more to say. But for today I'll just rather tell you slightly more about additional complexities of this system, and very briefly, because this is all based, next 2 slides, on purely results with recombinant protein in the tube, but they are very convincing. So I'm bravely kind of trying to tell you we believe something like this might be happening in vivo. So basically, the fact is that PAR14 is actually expressed from the same genomic locus, the same chromosome 3, like another PARP, PARP9. and delta X3L within ligase. So PARP9 is what we think is inactive PARP. It has a couple of specific changes in its catalytic domain that presumably makes it catalytic inactive, but it has this obligatory partner, which is E3 ubiquitin ligase with this ring E3 domain. So what we know, and our AlphaFold modeling suggests quite nicely that these three might go together as a heterotrimer in some cellular situations. We don't have ultimate proof for that, but what we know from, again, in vitro reactions, that actually this ring domain together with this DTC domain next to it, which is a novel type of ADP ribose binding domain actually. So DTC domain probably binds the arylated macromolecular targets and then allows C3 ligase domain of the DTX3L to ubiquitinate already arylated targets. So basically making a compound dual modification, quite novel paradigm. So if it's really exists in nature, it would be very interesting to pursue further. So basically, you have one modification on top of each other, linked to a target. And another thing I wanted to say in this context, if I bring you just briefly back to SARS-CoV-2 NSP3 full-length structure with this all multiple domains, actually you're well aware, some of you are at least well aware of PL-PRO, ubiquitin hydrolase domain, and its canonical function is surely in processing of the viral polyprotein into the smaller protein, like including NSP3, NSP1, and NSP2, cleaving after glycine residue in the polyprotein. But we have really good in vitro activity. I'm not showing the details. Most of it's recently published, but also, again, we don't have in vivo evidences. PL-PRO very efficiently cleaves these linkages as well. So altogether, it seems that you might have PARP14 deltax together pushing for this dual modification, and then two very neighboring components in coronavirus NSP3 can reverse it back in 2 steps. And again, these both domains are completely conserved in all coronaviruses. Many of them are lacking these 2 domains, so this would bring these 2 domains even in closer relationship. Yeah. So altogether, it looks like there must be very interesting biologies to studies to understand also as well, relevance for the drug targeting of these proteins. So, but at this stage, already we have enough genetic evidence, and many other people were encouraged to believe PL-PRO and surely MAC1 are promising drug targets. And for this reason, in the second part of my talk, I'll focus on these initial efforts to develop RL inhibitors for MAC1. So again, we started our efforts before pandemics, actually, trying to think it's important to target SARS-1 macrodomains and homologous macrodomains in other viruses beyond coronaviruses like herpes virus or some other alpha viruses. So they're all very similar, and the screening method will be exactly the same. But basically, we focus on MAC1 and do like multiple approaches in silico screening. Didn't yield much, so I'm not going to talk about it, but I'll tell you a little bit about our little in vitro screen that gave some good starting compounds, and as well a major effort with multiple collaborating labs, Frank von Delft, James Fraser, and Brian Sorkat in San Francisco, Paul Brennan, and many others. Fragment-based screen. So I'll tell you very briefly about these efforts. Most of this is published, so you can read details in the publications, these 2 publications, but I'll tell you a couple of points that I wanted to highlight, especially where we contributed more. So basically, focusing on this in vitro screen. So the key thing was the in vitro screen assay that developed several years ago, well before pandemics. It's a thread-based assay, HTRF technology-based screening assay, where we use short peptide, where on one position we have artificially linked on the side chain of the lysine ADP ribose via an uncleavable linkage. And in the proximity, we have also binding group that allows coupling to the HTRF acceptor. So on the other hand, we can have histag macrodomain, and this would allow the system to work and allow screening for compounds that will disrupt this interaction and diminish the the signal. So we did screen a relatively small library or 10K like variety library plus 2K of FDA approved drugs library. And just very briefly, we got several scaffolds that went to low micromolars. I won't go into details. Of FDA approved drugs, we picked up three of them. And I'll just pinpoint one hit in terms of antibiotics, just to pinpoint typically major interactions we are observing in the hits that we are getting through these approaches as well as fragment approaches. And this can be maybe visualized if this starts turning around from this little movie. So we have astranon antibiotic here. It binds to adenosine side roughly of the enzyme compared to ADP ribose bound, and typically we get this kind of hotspot phenylalanine 156, which we get in most of our interacting molecules. And also I would point that commonly our molecules have carboxyl group to interact with what we call the oxyanion part of the ADP ribose binding pocket. So this is just to show you one example to give you some feeling, and it will be easier to describe a couple of things in the next few slides. But I wanted to say a few more sentences on crystallographic fragment-based approach that we did, as I said, with massive collaborative effort. And the key for this approach for us is a bit of luck early on, where we were testing different cuts around MAC1 gene to get as crystallizable system, and with very specific cut, nothing left or right could be left longer or shorter. We get really nice chunky crystals. They grow super fast, go to super high resolution, 1 angstrom, and they are very soakable. So that was the main basis for the fragment screen, and then we shared these conditions with our collaborators at University of San Francisco, and joint effort to use our libraries together with Diamond and San Francisco libraries yielded, together with a few hits that came from docking approaches, yielded 254 crystal-bound fragments that were confirmed by in-solution activity assays, DSF, HTRF, which we use also hydrogen screen or ITC. So it was a really good start, really strong start for fragment screen, and then this was developed further. But before that, I'll just pinpoint again that in terms of hits, almost all of them nicely hit the active site, ADP ribose binding site, and within that, actually, most of the hits are actually around adenosines and adenine sites and oxyanion part of the pocket. Whereas catalytic site, we literally had almost no hits. Now, to again here to emphasize again, many of the hits were targeting this phenylalanine 156, which I immediately say is not conserved in most of the coronaviruses. Is usually replaced by asparagine, but in some human proteins still exist, like in macrodomain. Another very common hit is aspartate 22, which is actually very conserved and not beyond coronavirus. In fact, most of the human macrodomain proteins would have this residue conserved. So altogether, it looks like it should be relatively easy to make specific compounds, but also maybe some pan inhibitors as well. But anyways, I won't go into details of follow-up studies because it was not done by us. We just had provided supporting studies. But again, fragment linking approaches and fragment merging approaches were successful and eventually yielded promising compounds, and also ultra-large docking effort by San Francisco collaborators, which are focused on these hotspots of adenosine sites and oxyanion sites, and eventually again leading to low micromolar compounds. The best effort so far, I mean, from our collaborators, came from initial fragment linking and then purchasing analogs. And currently, there is the best compound that I'm aware of, 0.5 micromolar IC50. And we know for main hits noted here that they're already selective over human macrodomains, TARG1 and macroD2 that we routinely used as a counter screen. But again, in terms of further selectivity, because these novel discoveries of PARP14 macrodomains and even PARP9, they're actually closer. overall, we can already say as well for them, specificity shouldn't be a problem. This phenylalanine is not conserved in catalytic macrodomain of par14, for example. So altogether, we didn't test again specificities for these targets. We'll do soon, but again, if need be, selective compounds will be easy to do. And this is just a summary then of the effort. So basically, by starting altogether at the end by several hundreds crystal structures, we are now with this effort we are down to very low micromolar activities, and of course, next step would be improvement of cell permeability for antiviral studies. Yeah, but other people will update you on that. So coming to conclusions of my talk, I don't know if I'm over time or not, but I'll still say the main conclusions. So basically, we identified par14 as the main source of interferon-inducible hydrophosphorylation in cells. We don't know exact mechanistic effects of that. There is some suggestion that it would affect interferon signaling in cells or some other aspects, but it's not clear. We also identify MAC1 as the main reversing enzymes in cell for this signaling. Again, mechanisms need to be clarified in the coming years. But at the same time, as I said, the complexity will grow almost certainly because we know that arypsilated target will likely be ubiquitinated in cells, making this compound modification that again could be another level of warfare, whereby then virus needs to have other ways to counteract this. And at the end of my talk, I showed you the MAC1 inhibitor development strategies are underway and they look promising. And also throughout my talk, I indicated potential alternative drug targets in the pathway, maybe even par14 macrodomain 1, because it suppresses maximum activity of par14, so blocking that might make sense in its own right, or dual inhibitors targeting both. I also told you in the beginning of my talk that PARP7 is actually has proviral function, so maybe targeting its catalytic transferase domain may make sense, but also other targets, PL-PRO, PARP9, delta-XL, who knows? So altogether, promising field worth exploring, and I'm happy, to answer any questions now or collaborate in the future. Yeah.
Thanks so much,
now,
Ivan.
Most important slide, I can't read everybody because there's so many people that were involved in here. But you can read them, and I'm grateful to everyone. And it was a fantastic joint effort, on all aspects. Yes, thank you.